Hello everyone, hope all of you are fine. So, dear children, we are doing cell cycle and cell division. So, in this part, we'll do the meiosis-II and one part that is interkinesis or intrameiotic interphase. Okay, and significance of meiosis. So significance of meiosis meiosis one we had discussed, and in this part let's discuss about the interkinesis or intermeiotic interphase. Okay, it is metabolic stage between the telophase of meiosis 1 and prophase of meiosis 2. Chromosomes are elongated but chromatin reticulum is not formed. Protein and RNA synthesis may occur. Centrosomes or centriole pairs undergo replication in animal cell. However, there is no DNA synthesis. It is important for bringing true haploidy, that is haploidy of DNA in the daughter cell. Now we will discuss the meiosis-II. Meiosis-II, it is shorter than the typical meiotic division, mitotic division, because of the shortening of the prophase of this division. The division maintain the number of chromosomes produced at the end of the reduction division. It is hence called homotypic or equational division. Though it is similar to mitosis, meiosis-II is not mitosis because it always occur in haploid cells. It is not preceded by DNA replication. The two chromatids of chromosome are often dissimilar. Thus, daughter cells formed after meiosis-II are neither similar to each other nor similar to the parent cell. The main function of homotypic division or the meiosis-II is to separate the chromatid of the univalent chromosome which differ from each other in their linkage group due to crossing over. Meiosis-II is divisible into prophase, metaphase, anaphase and telophase. So prophase 2, this stage of nuclear division is very brief. It takes place simultaneously in the two nuclei. In animal cell, the centriole pairs develop aster and move to the region of future spindle pole. The dyad chromosome shorten a little, nucleolus and the nuclear envelope degenerate. The chromatids of individual chromosome are usually divergent and hence much looser than ones in the somatic division. In case where telophase 1 is omitted, the prophase 2 is completely absent. Now next we'll do the metaphase 2. According in metaphase 2, achromatic or achromatic bipolar fibrous spindle are formed in the areas of dividing nuclei. They are amphiastral in case of the animal cell and anastral in case of the plant cell. The spindle are arranged in isobitral or bilateral or tetrahedral fashion. The chromosome reach the respective spindle and arrange themselves in such a fashion that their centromere come to lie at the equator. Each chromosome gets connected with both the spindle poles by means of chromosome fibers or tactile fibers that develop from both the surface of its centromere. The centromere give out chromosome fiber or the tactile fibrils from both their surface towards the spindle pole. Now next we have the anaphase 2. So in anaphase 2, the centromere of each chromosome divide into two so that there is one centromere for each chromatid. The two chromatids of the chromosome separate completely and are called daughter or new chromosomes. The daughter chromosomes moves towards the spindle pole along the path of their fibers or the tactile fibers or fibril. At the end of anaphase 2, four groups of chromosomes are produced, each group having haploid number. Now next is the telophase 2. Telophase 2, the Four groups of chromosomes arrange themselves into haploid nuclei. For this, chromosome elongate very much to form chromatin. The nucleolus is also produced. This is followed by formation of nucleoplasm and a nuclear envelope. The spindle fiber usually degenerate during the telophase 2. Now next is the cytokinesis. Now cytokinesis include the division of the cytoplasm. Okay, cytokinesis can be of two types, successive and simultaneous. In successive type of cytokinesis occur after every nuclear division. It produces two cells after the heterotypic division. Both of the cell undergo homotypic division and then give rise to four cells. The four cells formed by the successive cytokinesis can be arranged either in a linear or isobilateral tetrad. The in simultaneous type, the cytokinesis take place only at the end of both of the division. The nuclei are generally arranged in the form of tetrahedron. The cytoplasm cleavage or constrict in between the nuclei. Four furrows are formed. They deepen and meet in the center of the parent cell. The plant wall material is deposited in the furrow. It gives rise to four haploid cells. These haploid cells are arranged tetrahedrally and are collectively called tetrahedral tetrad. Okay, now we have the significance of meiosis. Significance of meiosis: formation of gamete. Okay, meiosis form gamete that are essential for sexual reproduction. Genetic information: it switches on genetic information for the development of the gametes or gametophyte and switches off the sporophytic information. Then it also help in maintaining the number of chromosome. Okay, meiosis maintain the fixed number of chromosome in sexually reproducing organism by halving the same. It is essential since the chromosome number become doubled after fertilization. Assortment of the chromosome: in meiosis, paternal and maternal chromosomes assort independently. It causes reshuffling of the chromosomes and trait controlled by them. Or variations help the breeder in improving the race of the useful plants and animals. Now, meiosis also introduced new combination of traits or the variation, that means crossing over take place in meiosis. Mutation: chromosomal and genomatic mutation take place by the irregularities of meiotic division. Some of these mutations are useful to the organisms and are perpetuated by the natural selection. Evidences of basic relationship of organisms. Detail of meiosis are essentially similar in the majority of the organism, showing their basic similarity and relationship. Okay, now why the meiosis is needed? Okay, meiosis is essential for sexually reproducing organism. It occurs in reproductive cells so that the gamete formed are haploid or half the number of chromosome of those cells, which are directly derived from the zygote. The two type of gamete fuse during the zygote formation. As a result, zygote come to have double number of chromosome contained in the gamete. Meiosis, by halving the number of the chromosome, maintain a fixed number of chromosome of the species. In absence of meiosis, the number of chromosome will double with every generation, resulting in the excessive enlargement of the nucleus, genetic degeneration and death of living beings. Now, types of meiosis. Okay, the cell in which meiosis takes place is called meiocytes. In animal, meiocytes are of two types: spermatocytes and oocytes. In higher plants, meiocytes are differentiated into the microsporocytes and the macrosporocytes. Depending upon the stage when meiosis occurs, the latter is of three types, that is gametic, zygotic and sporic. So you can see in the diagram. Gametic meiosis. Meiosis is the first of animal take place during the formation of the gamete. Okay, that is gametogenesis. It is termed as gametic meiosis when two gametes fuse in fertilization. A diploid zygote is formed. Okay, gametic meiosis results in the diplontic life cycle. Now next we have is zygotic meiosis. In some lower plants, meiosis take place in the zygote and the resulting organism are haploid. Okay, it is called the zygotic meiosis. Okay, organism having zygotic meiosis are having haplontic life cycle. Next we have is sporic meiosis. In plant, meiosis generally occur at the time of sporogenesis, that is formation of spores or the microspores and the megaspore. It is called sporic meiosis or intermediate meiosis. Spores produce a new gametophytic phase in the life cycle. Gametes are formed by the gametophytes. Because of presence of two distinct multicellular phase, diploid and haploid life cycle of the plant is diplontic. Now diplohaplontic. Okay, diplohaplontic, that means diploid and haploid both phases exist in their life. Diploid cells have two genomes while haploid cell have a single genome. A genome is a complete and complement or set of chromosome where each kind is presented by a single chromosome. Now let's do colchicine. Okay, it is an alkaloid widely used in the plant breeding for doubling the chromosome number. Colchicine is extracted from the corms of autumn crocus, that is Colchium anumuneli. Okay, it is Colchium autumnale. Okay, the alkaloid does not allow the formation of spindle because it prevents the assembly of microtubules. It is therefore called the mitotic poison. Okay, colchicine is also known as the mitotic poison. The chemical does not inhibit the chromosome replication. As a result, the colchicine tetrad meristematic cells show doubling of the chromosomes. This property of increasing the number of chromosome sets or genome is called the polyploidy. Polyploidy provide new variety and species vigorous offsprings. Colchicine induce polyploidy has been used in raising several varieties of horticultural and agricultural plants, for example, potato. Now let's discuss some differences between the mitosis and meiosis. Mitosis, it takes place in somatic cells. Meiosis, it occur either in the reproductive cell or at the time of the germination of the zygote or zygospore. The cell undergoes mitosis, may be haploid or diploid. Okay, mitosis can take place in the haploid or diploid organism, and the cell undergoing meiosis are always diploid. Okay, meiosis take place only in diploid cell. It is a single division which produce two cells. Mitosis produce two cells, meiosis produce the four cells. Okay, and it meiosis is a double division. Okay, interphase occur prior to each division, that means before the division the interphase is there, and interphase proceeds only meiosis 1. It does not occur prior to meiosis 2. Mitosis is comparatively simpler. Okay, it is simple, and meiosis is quite complicated. It takes less time to complete, that is mitosis, and meiosis take long time to complete. Then subsequent mitotic division are similar to earlier ones, and the two division of the meiosis are not similar. Okay, the first one is heterotypic or reductional, while the second one is homotypic or equational like mitosis. Now each chromosome replicates in the interphase before every division, and in case of the meiosis, the chromosome replicate only once prior to meiosis. The number of chromosome remains same after mitosis. The number of chromosome reduces after the meiosis. Okay, the daughter nuclei or the cell formed after mitosis are exactly similar to the parent one. The daughter nuclei or the cell formed after meiosis are neither similar to the parent nor to one another. Next we have meiosis help in multiplication, meiosis help in the multiplication of cells. Multiplication of cell is not involved, so mitosis, it will help in the multiplication of cell. Okay, and mitosis take part in healing and repair. Meiosis take part in the formation of neospores or the gametes and maintenance of the chromosome number of the race. No variation are introduced in the mitosis, and meiosis introduce variation. Okay, so prophase of the mitosis, chromosome are not conspicuous. In meiosis, chromosomes are quite conspicuous. Prophase is of short duration in mitosis. Prophase 1 is longer duration, while the prophase 2 is very brief. The prophase is simpler and hardly distinguishable into the substages. Prophase 1 is complicated and divisible into 5 substages. The prophase 2 is, however, very simple. Each chromosome has 2 distinct chromatids. Okay, the chromosomes of prophase 1 do not show distinct chromatids. The no bouquet stage is recorded. Okay, bouquet stage is not recorded in the mitosis. In the chromosome of the animals and some plants show convergence towards one side during early prophase 1, and it is known as bouquet stage. Now pairing of chromosome does not occur in the mitosis. Okay, and pairing of the synapses of the homologous chromosome take place during the zygoteny of the prophase 1 and continues up to metaphase 1. As synaptonemal complex is absent, as synapsed homologous chromosome develop a synaptonemal complex in case of the meiosis.